over, so like my last final is tomorrow. Well, I only have one main final, which is tomorrow. That's genetics, but
Huh?
yeah, I'm just like transitioning to no life MCAT mode right now.
Are
Um,
you ready for genetics?
yeah, I am.
Hmm.
Uh, I'm not. I'm doing fine in that class, so the final, like even if I do poor, I'll still be good. So
Got it.
yeah. Okay. First of all, that textbook you sent me is really good.
Yep. 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 Yep.
I it it was really helpful. I mean, I won't fully know until I get an actual immune system question, but like it's I understood a lot of stuff
Mhm.
conceptually. Anything that I don't get in terms of physiology, like the cardiovascular system, I feel like I need a little brush up on. Should I read the textbook?
Yeah. 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 That's so yeah, like so when I was studying what I typically would do is um and I also have like three monitors. So like on one monitor I would have like an MCAT book, like the Berkeley Review is a really, really good one. And on another monitor I would have like a textbook. So the Saladin one is excellent for anatomy and physiology. There's also like Leninger's book on biochem, Lodish, I think book on Black Cell, Gian Coley's book on physics. So like I would have a textbook also open, and then I would have on my third monitor like Google image or Google. And so when would learn something, I would kind of like cross-reference it from all the different sources so that I get like the definitive answer. So and yeah, that's what I did with that book being an example. So for you, I think the workflow would be because I noticed when you were telling me about like how long it took to like watch like a video or something, and I was like, all right, like I know that you prefer watching videos and your generation prefers watching videos, but it is, a reading test, and you saw in that excerpt, right, that where I underlined like processing information. What I'm talking about?
Yes.
Yeah. So that's literally like that's a paper that's also in the shared drive. Like it's literally like a published paper on like MCAT tips. And yeah, it's processing information is a skill that can take years to develop. The best way to strengthen the skill is by simply reading as much as possible. So like sure, even if you I mean, ultimately, it doesn't even really matter what you read. Like, obviously, it's good to read like things that are going to be close to like the type of level of reading that's required on the MCAT, at least for cars. But like, if you just like try to read as much as possible, like, I don't know, like, so like for me, like, I'm reading things like randomly on Wikipedia. I'm reading things like randomly, just, because I'm wondering, I'm thinking about yeah, I'm like curious about things. So like I've always just kind of like read a lot. Um, it doesn't necessarily mean like, like I'm reading like a lot of books or anything, but just reading. Whatever it is, reading, like that's gonna be good for you. Um, so yeah. And we can obviously like make that maximize that by having you read like really, really good stuff that'll be similar to the stuff that you'll see on the MCAT, but yeah, at the end of the day, as long as you're reading, you're good. And yeah, so I guess the workflow would be like okay, so like let's say like immune system, and then you can use the Saladin book or one of the books there that I have in textbooks section, and you can kind of read up on it like that, and then you can use the like BB content, CP, like, those tabs that I made in the spreadsheet to like see exactly what you need to know and how much you need to know. And then you can just update that from like weak to okay. And then, after you do some more like problems and questions, like as you said, for the immune system, like, you just have to do like questions and stuff to really see if you can go from okay to like I forgot what like the last one was, but it's like good, I guess. So that can be the workflow. Um, and that means that you're going to be reading, so you're going to increase your improve your reading skills. And it should take less time. So it should, uh work in that regard too. Because yeah, well, I was thinking about it and you were saying like, it takes a lot of time and then if it's a video, like yeah, it'll take as long as that video length is, right? And I mean like if you do two times speed, it'll be half that length, but I still think it'll be longer than if you like had read it. So yeah, let's start doing that for like a topic that you want to like look into and stuff. What do you think?
Yeah, it's fine.
Is
I'll
it
do it.
like difficult reading through that uh chapter?
I mean, I had to read a couple things. Like,
Uh-huh.
what one issue I just have in general is I don't know. I mean, I'm not diagnosed with ADHD or ADD, but like, just when you're trying to read something, but then you're thinking about something else.
Yeah,
So
yeah.
you just like, glance over it, so you go back and reread it, and that, spends time, wastes time.
Yeah,
I
yeah.
had to do.
Yeah. So I would say here, let me go to like the um
I do have to leave at like 7, so let's try to make this like efficient. But
yeah, yeah, yeah. Um, but I also don't want you to think like the only way to get things done is by, getting X number of, questions,
yeah, yeah, yeah.
stuff
I
like that.
realized that.
So, but I was just pulling up the chapter. And so like right away, like functions of the lymphatic system, immunity is one of them. But the other one is lipid absorption, and that's good to know because they'll people don't really talk about the lymphatic system that much, and that's something that happens there. And then for like the immune part, you got to see like I guess like the broader examples of like lymphocytes and stuff and antibodies. And then you saw like the stuff about like the granulocytes and everything like that. Anyway, yeah, I wanted you to read it without taking notes or anything like that because I wanted you to get a big picture view of it rather than getting, too much into the details. So yeah, I think that this could be a good workflow and uh, and yeah, and from like the flows, yeah. So like, and from the flow stuff, you see that, you want to know how do you know you're like, so okay, you talked about how like, it's if you're reading something, it's kind of you can get distracted and think about something else, then go back to reading it. But like, what about when you're feeling like in the zone? Do you know what I mean? Like in the zone.
Yeah, I mean, then I'm not doing that as much, right? Yeah.
Oh no, no, I'm just curious, what like what are some experiences that you've had where you're in the zone or something like that? Um, like,
Um,
huh?
I mean, I don't, I guess I'm like a lot more focused on, it also depends if I find the topic interesting.
Yeah, yeah,
Like,
yeah. So that's one of the things in the flow thing. Like you find it intrinsically interesting. So that's why I think I forgot if I wrote this, but like with the lymphatic system, it's like, or the immune system, it's like you're going to be a doctor. You're going to need to know this stuff, right? So it could help to be like interested in it, and it can also help to like learn the stuff without thinking, okay, this is what I need to know. Like, you don't want to be like, okay, I'm learning this because I need to in order to be a doctor, right? Or I need to in order to do well on this test so I can become a doctor. But if you look at it as intrinsically rewarding, like I'm going to learn this because I want to be like, the best doctor I can be. So I want to learn. You want to do more than just like the bare minimum, right? And then also just in general, if you can convince yourself to be interested in learning anything and everything that comes, across your path, that's just a very, very good attitude to have. So, these things lead to those flow states. So yeah, so I would say when you're doing something, don't think, you know, am I wasting time? Am I don't yeah, try not to think that way and try to just like completely engross yourself in what you're doing. And signs that you're in a flow state will be like that uh, loss of viewing yourself from the outside. Like if you're viewing yourself from the outside, it's like, okay, am I wasting time here? But loss of that type of like awareness and kind of being just really like in the zone, and everything that you're learning you find to be intrinsically rewarding. So um like yeah, finding we'll find ways to like make that, more of a thing. But yeah, I would say most importantly, try not to think about whether something is worth it or not, depending on if it's a waste of time or not a waste of time. Anything that you do that's MCAT, prep is worth the time. So, and it's quality over quantity. So I guess that's like just my closing kind of statement there. So
Mhm.
yeah. So anyway,
Okay.
what would you like to do today?
Um, this passage. Do you see it?
Hmm.
Yeah.
Now, have you done this before?
Yes.
Okay. So, uh, take the reins, read it. Tell me what you're thinking as you read it. Um,
Okay.
I'll kind of like stop you like at random points for like buzzwords and stuff, but also if you recognize buzzwords, tell me that as well. So yeah, why don't you go for it?
Okay. Fatty acid synthase is a key cytoplasmic enzyme responsible for synthesis of long-chain fatty acids.
Okay, tell me.
Synthase, it's a type of enzyme.
Good.
Now, we went over this last time. There's a difference between synthase and synthetase. I think synthase doesn't require ATP.
Uh, it's okay. We can just say, okay, this is a synthase, right? So it's an enzyme, and what does it do?
It creates fatty acids.
Uh, just fatty acids?
Oh, you're asking about synthase in general?
Oh no, no, just here. Well, in this first sentence,
Long-chain fatty acids.
long chain fatty acids. All right, and there's one. So you see, okay, fatty acid synthase enzyme. It synthesizes long chain fatty acids, so that's important. There's one other thing that's important here.
Cytoplasmic.
Exactly. Good. Continue.
Abnormal fatty acid synthase expression is associated with tumor cell progression in human cancer cells, and as such, FASN is being investigated as a target for cancer therapy through development of specific inhibitors such as C75, a substrate analog.
Okay. They said abnormal FASN expression cancer. Now, do you think that so abnormal can mean overexpression? It can also mean underexpression. What do you think it would be here?
Over. Wait, wait. I think tumor cell progression. I would say over, yeah.
All right, tell me why you think it would be over.
I mean, tumors, there's usually overexpression in tumors and
Well,
cancer.
well, uh, you can logically answer it.
Well, you're inhibiting it. I don't know. Or maybe that the cells use fatty acids.
So, so you were saying like, okay, tumor cells usually have more expression. That's some type of like perhaps memory like content thing, but logically we can see, right? Because let's try one more time. So like logically, why do you think FASN like overexpression would be associated with tumors?
I mean, when you're overexpressing something, there's more of it.
Well, what if it was like fatty what if it was lipase?
Mm.
The overexpressed lipase you'd have less.
Yeah, less fatty acids.
So now the thing is that this is responsible for the synthesis of the long fatty acids, and cancer is when you're making a lot of stuff, right?
Hmm.
So FASN makes stuff. So it would make sense that if you have overexpression of FASN, you'll be making so much stuff that, and cancer loves making stuff.
Okay,
Yeah.
that's it.
Yep.
Okay. In addition to the FASN pathway, a
Oh, sorry.
mitochondrial.
Sorry.
Yeah,
What type of inhibitor do you think C75 is?
competitive
How do you know?
substrate.
Substrate what?
Like, um, it's substrate analog
Yeah. What does that mean?
that like it fits into the active site as like a substrate.
Yeah. It basically is like an analogy of some sort. It's like the equivalent of a substrate. So good. Very good. All right, continue.
In addition to the FASN pathway, a mitochondrial fatty acid synthesis pathway has been identified.
Okay.
Um, okay. The key enzyme of this pathway is the mitochondrial acyl carrier protein synthase, MTCAS. MTCAS is also involved in the synthesis of lipoic acid, a cofactor for several metabolic enzymes.
Okay. So we know that MDCAS. Oh, I didn't even write any proportionalities yet, but MDCAS. Uh, well, actually, so okay. What proportionalities can you write? Starting from the top. Let's start with FASN.
You can, I mean, you could technically write that it's associated with tumor cell progression, so like it's proportional directly to that.
Yeah, I mean, we could start with saying long chain fatty acids,
Oh yeah.
and and then sure, you could say like whatever cancer, and then maybe inversely proportional to C75. And then we have MTCAS should be proportional to what?
LA.
Hmm? Good. Continue.
The deletion of the gene encoding for MTCAS results in dissipation of the electrochemical gradient in mitochondria due to an increase in membrane permeability.
Okay, tell me about this.
So I was a little like I basically it's saying there's an increase in membrane permeability. So I would think that like basically more it's easier for things to go through the membrane.
What membrane are you talking about?
The inner mitochondrial, I mean the mitochondrial membrane, sorry.
Wait, which one though?
Like the one inside of the
Yeah.
mitochondria.
Right. Now, what's special about that? What happens there?
The ETC happens there. So like protons get pumped out
Yeah.
into mitochondrial space and then pumped back in.
So, tell me a little bit more about that. What do you mean pump back in?
Um, they get pumped out through the
Yeah.
whatever they're called.
Yeah, maybe that.
Through one, three, and four. Um, and then they get pumped back in, and that's what allows for oxygen to um to get reduced, I think, and create water, and water is created. But that creates ATP.
Uh, different, slightly different here. So, uh, it's okay. The complexes you mentioned,
Complexes, yes, yes, yes.
and you were saying, what about the complexes?
Basically, that different electron carriers,
Hmm?
NADH and FADH2, they
Yep.
go to the complexes and they donate electrons that pumps out hydrogens,
Okay.
and the hydrogens get brought back into ATP synthase, and that creates ATP.
Okay, okay, okay. So, because you said something about oxygen.
Right, and oxygen is it accepts electrons.
So that's at the end of the ETC, right?
Okay, okay.
But but yeah, that's what happens, and it pumps protons into the intramembrane space, right? And then uh you said that they get pumped back in. So if pump by pump, what we generally mean is like um against concentration gradient. So do they get pumped back in, or does something else happen?
Oh, true. Well, I mean, because
Hmm.
they're like at a high concentration, I guess they
High concentration
go high concentration outside
outside where?
of they're in the mitochondrial they're in the
Uh-huh.
mitochondrial membrane. Like they're between the outer mitochondrial membrane and the inner.
Yep, yep, yep.
That's where there's a high concentration and they go back in.
Yeah, yeah. So good. So you have a higher or lower pH in the intramembrane space.
Lower.
Good, good, good. All right, now, and so they naturally go down their concentration gradient as they go back into the matrix, right?
Yeah.
Because, yeah. So, and that gets coupled to the ATP synthase to make ATP, right?
Mhm.
So if you and so the inner mitochondrial membrane is not permeable, but if it was, what would happen? Like permeable to the protons.
It's not permeable to the protons.
Uh-huh, right?
Okay.
Meaning because if it was because the complexes pump the protons out and then they have to go in through the ATP synthase. So that means they can't just go right back into the matrix, right? That's what I mean by
Yeah, but the thing is, what I didn't understand when I was reading this, like I knew they'd ask me about something about permeability, but like can't if there's increased membrane permeability, can't it go out of the membrane just as equal likely as it can go back in through the membrane? Like, you know? Because it goes out through the complexes, from what I understand, and it goes back in through ATP synthase.
yeah, yeah, yeah. So the it'll go down its concentration gradient. So if we have like the ETC stuff here pumping protons out, right? It means we have more protons out in the inner membrane space than in the matrix. So if you, and then you have your ATP synthase, so it goes down and it can make ATP. So if you made, so this means that it's not permeable to the protons, but if it was, the protons can just go right through that to come back to the matrix because the ETC pumps them out, creating a concentration gradient where in which the we have a higher amount of H plus here and a lower amount here.
Mhm.
So if the membrane, inner mitochondrial membrane was permeable, the protons would just go from high to low until it's equilibrium, same amount on both sides, right?
Mhm.
So, so that's why the it's not this inner mitochondrial membrane is not permeable. That's why it would have to go through the ATP synthase. But if it was permeable, it can just go down, like go through the membrane, right?
Got it.
And if it goes through the membrane, then it'll bypass the ATP synthase and you won't get ATP made.
Mhm.
You know what I mean? So I think your question is so if the increased permeability means that it can go into the matrix, you're asking can't it just go out of the matrix as well?
Yeah.
Yeah. And so the answer to that is just that the ETC pumps it out, right? So you have a higher amount in the inner membrane space. But if it was permeable, it'll go back down until it's at equilibrium, right?
Okay.
So, I mean, let's say theoretically we created a high concentration of H plus inside and a low on the inner membrane space, and we had permeability, then it would go that way. But it's really just going to go down its concentration gradient.
Okay. Got you.
Yeah. So they like to ask this type of thing. Like I've seen it many times in different contexts, like brown fat, thermogenesis, 24 DNTP, no, 24 DNP, um, are all things that basically decouple the proton gradient from the ATP synthase. So it's good to have a intuitive understanding of this. And you see how it says dissipation of the electrochemical gradient? This is the gradient, high here, low here, right?
Mhm.
Okay. So, um, what can we write here in terms of proportionality?
Um, I don't know. Hold on. I mean, MCAS,
Mhm.
um, is proportional to 1 over ATP.
Hmm. Where do you see anything about that?
The thing is, like, if there's an membrane permeability, there would be less ATP made, right? Because they could just go through the membrane, the H's.
Oh, sure. But you said empty cas.
Well, you delete it.
Uh-huh.
So, like, technically, so I mean, okay, you could say 1 over MT cas is like proportional to, um,
We could maybe do this. Usually a clue with proportionalities would be like something goes up, the other thing goes up, something goes down, the other thing goes up.
oh, so increase in maybe membrane permeability leads to dissipation of electrochemical gradient.
Right? Increase in membrane permeability, MP, dissipation or decrease of electrochemical gradient, 1 over EC gradient, right?
Mhm.
So you see how we're looking at these keywords, right?
Okay.
Okay, continues.
So the dissipation of electrochemical gradients, just double check, that's like it means there would be less of like high to low or low to high or whatever. Like it would be more.
So electrochemical gradient, right? So gradient means like a difference, and
Mhm.
so many things can be expressed that way. For instance, a battery, right? An electrochemical cell, right? There has to be a gradient, meaning like a positive negative there, right? There's a gradient here, right?
Okay.
But when the battery is at equilibrium, there's no more gradient, right?
Okay.
So gradient is the difference. Um, so yeah, if there's high in one compartment and low in the other, there's a gradient. Um, if you dissipate the gradient, so there's a gradient here. If you dissipate one, it'll be like that
Mhm.
with no differences. Equilibrium. So if you have same H plus constant or same pH in the inner mitochondrial matrix and the intermembrane space, will you have a electrochemical gradient?
What?
So let's say pH of the intramembrane space is equal to pH of the matrix. Is there,
There's no electrochemical gradient.
you said there's no electrochem?
Yeah.
Good, because they're the same.
Yeah.
Good.
All right. Since MCAT shares conserved catalytic domains with F and that are involved in binding to C74, 75, researchers investigate the effects of C75 and MCAT's function.
When I say conserved, what do they mean?
Um, Maybe similar.
So that generally means that they kind of like co-evolve.
Hmm.
So they're saying FASN has like similar evolutionary path uh with empty cas, and because of that, it could be the case that C75 works for empty cas just as it works for FASN.
Okay.
Yeah, so when you see conserved, think about that.
Okay.
All right, cool. So we wrote our proportionalities, and yeah, let's go here. Experiment one.
Uh, T93 T cells were treated with three different concentrations of C75, and inhibition of MT-CAS was analyzed by measuring cell viability and mitochondrial membrane potential as a function of time.
Okay, now mitochondrial membrane potential.
Uh-huh.
Tell me about that.
S- Similar to what we were just talking about
Oh,
with electrochemical gradient.
yep. So in this situation here, right, is there what would the well, it would be a voltage or a potential as well, right?
Yeah,
So tell me about the potential for this compared to this.
the potential in the one on the left, there's a existing potential, and the one on the right, there's none.
Absolutely perfect. Good. So now, what about proportionality between membrane permeability and MMP?
1 over MPP,
Well, sorry.
1 over MMP.
Yep. Membrane permeability, less of the gradient, less of the MMP.
Uh-huh.
Good, good, good. Okay. And then cell viability, like that's just what do you like? What's that?
If the cell is viable, meaning if the cell is like able to survive.
Yep. Okay. Good. So we see here cell viability, black bar control, and then the other ones, just get larger in concentration. What do you see for A?
Over time, when you have an increase in the concentration of C75, um, increase in concentration leads to less cell viability.
Okay.
Um,
You do a proportion here.
yeah, so cell viability. Well, you could do, yeah, C75 is proportional to 1 over cell viability.
Any caveat?
Yeah, I mean, it increases in 2 hours.
Well, we kind of don't see much in that 2 hours, much of a change, right?
Okay,
So maybe at equal or greater than 12 hours.
okay.
And you see how, oh, why do you think I put brackets around the C75?
That means concentration.
Yep. All right. Next.
Um, in MMP, also, as you increase concentration, there's decrease in MMP.
Any caveat?
Mm, like after 2 hours and over up.
Greater than or equal to 2 hours, right?
Yeah.
Okay, cool.
Um, next, cells are treated with siRNA to knock down F FASN and TCAS, or both, and cell viability and MMP were measured.
Okay.
Okay. They used siRNA. They knocked down different, I guess, genes that express these enzymes. Yeah,
Yep.
or enzymes and or whatever, co-competitive inhibitor. And yeah, wait, no, they didn't do it 75. But yes, okay. Anyways, that's what they did.
Yeah. What do we see here?
Uh, well, when you had basically when MTCAS was present, there wasn't like that much of an empty like decrease in cell viability. But when MTCAS was removed, basically when MTCAS was removed and there's only FASN, that's when there was the least cell viability.
All right. Well, can we write in proportionality?
You can write FASN is proportional to 1 over cell viability.
Hmm.
What?
Uh, you said fast n is inversely proportional to cell viability.
Yeah.
Um, let's see. When you get rid of fast n, cell viability doesn't really change much, right?
But FASN is proportional to 1 over cell viability, no?
So without so wait. So you're saying um
FASN is proportional to 1 over cell viability.
okay. I think that so the thing is that with fast n, we don't see much of a difference. Like between these two bars, like there isn't much of a difference here.
So we're
But
silencing it.
so if you get rid of fast n,
Mhm.
don't have much of a difference in cell viability. So there's no
Okay, yes. Okay, then empty cast.
empty cast. Yep.
Okay.
So
Yeah.
empty
Yeah. Yeah.
gas, if you get rid of that, you have less cell viability, right?
Yep. I
So
got confused. I was wrong.
gas
Yep.
could be proportional to cell viability, right?
Okay.
So yeah, be careful with the siRNA thing.
Yeah.
So I mean, but you got it right the first time.
Right.
So okay, and then let's look at this one.
Yeah. Here, I guess you can write the same thing, but like for MMP.
All right, cool.
All right. So now figure one. Can we remove the blue, by the way?
Oh yeah,
Yeah, I'm getting OCD.
yeah.
But yeah, what conclusion about the data shown in figure one is valid? So here, like, when I saw these choices, um, I was wondering if we have two choices that are fully inverse, does that mean that likely one of them is correct?
That oh well, I would say it depends on the situation. But in this case, I would say it's healthier to not think of it that way.
Okay. A, changes in MMP likely affect cell viability. B, changes in cell viability likely affect MMP. Um, so like at first I didn't even think that like we can make conclusions on how like two different dependent variables affect each other. So when I was doing this, I didn't think either were correct. But then I realized the whole entire question talks about the relationship between two dependent variables. So I mean, MMP is unrelated to cell viability. That's what I put at first. That's what I put because like when I looked at 2 hours, I saw there was nothing happening, but MMP it decreased as in there was an increase in concentration.
Tell me about these first two choices.
Oh, um, so I mean between those two,
Just A. Like what is that saying?
saying that when you have a change in the
Hmm.
mitochondrial memory potential, can lead to affect cell viability.
So you
I
said
guess.
lead
Hmm?
you said can lead to.
Hmm.
Okay. That's significant.
Yeah.
So if changes in MMP can lead to changes in cell viability, right? That's saying something. That's saying so like the likely effect part is not helpful, but when you said leads to, what's that? What's the significance of that word?
Causal.
Yeah, yeah, exactly. Perfect. So let's see, right? So yeah, what do you see?
Well, at 2 hours, cell viability is still pretty constant,
Hmm.
but MMP is already starting to decrease.
Okay.
So if I had to choose between A or B, I'd choose B.
Hold on. S Say what you just said before.
Actually, what do you mean? Say what? That MMP is starting to decrease at 2 hours?
Good.
And I guess that yeah, then cell viability starts to decrease.
After 2 hours.
Ah, okay. Yeah,
So one
yeah.
comes before the other, and you said leads, so you understood that part, right? So,
Wait, but the leads is not the same as effects, or it is likely effects.
so effects is super vague. So,
Ah, okay.
but what you did was you said leads, and
which
that
is not good.
no, that's so that's exactly what you like. You're doing that perfectly.
Mhm.
Then you see that it's causal, and then you can see, okay, if something causes the other thing, the thing that causes the other thing, like if A causes B, A must happen first, right?
Yeah, yeah, you're right there.
So what
Yeah.
do we see here? What happens first?
MMP changes,
Uh-huh.
and then solubility then changes.
Yep. So would that be closer to A or B?
Hey,
Yep.
yeah.
But
C,
you did a good job there. Oh yeah. So um unrelated,
yeah.
MMP is unrelated to cell viability. I mean, eh, if that was the case, I don't think we would see these types of results here. Uh,
Mhm.
like these consistent results. Um, and then the cell viability can be restored by decreasing MMP. When we decrease MMP, it seems like cell viability also decreases. So yeah.
Okay. All right, next. Just because time is
Yep.
not of the well, time is of the essence. Sorry. Here I want to skip this. They talk about radical scavengers. The answer was reactive oxygen species. Like, I don't know. I I just want to skip it. If we could get to it, get to it. But that one I got right. Here, this was the big one that I really want to go over.
Okay. But
Uh,
just the stuff that we were doing, like I want you to see that that was important. Like you being able to think it through like that. So just saying.
yes.
All right. So okay. So yeah.
Cool. Which control experiments should be included in the design of experiment two to validate the results shown in figure two? One, evaluating the protein levels of FASN and MTCAS following siRNA treatment. Wait, experiment two. Valuing the protein levels of FASN and MTCAS following siRNA treatment.
What is this question asking us?
Which control? What control experiment should we do? Like? I mean, it's asking for like controls.
So like tell me like exactly what it's asking because we want to make sure we understand it. Don't worry about time here. Like I know you're worrying about time and that you want to get through the questions, but how you get through the questions is going to be more important. So let's see what is it asking us, right?
It's asking basically what is a control experiment that we can set up to make sure that um it, that the siRNA is targeting specifically that gene, which leads to whatever, like instead of targeting something else.
Oh, so yeah, in jet. So I think you're doing like one of the Roman numerals, but yeah, the question's asking what control experiments can we do to validate these results, to make sure that these results are valid,
Mhm.
right? So that's yeah. So now if we look at Roman numeral one, it says evaluating the protein levels of FASN and MTCAS following siRNA treatment. What are your thoughts there?
Um, I don't know if that would directly tell us like I don't know if that would directly tell us what whether or not it leads to, survivability or MMP. Wait.
Well, so 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 figure two
Hmm.
only tells us about MMP and cell viability, right?
Okay.
And it also tells us about the siRNA for each of these things, right?
So
Uh-huh.
yeah,
So we want to know what doesn't it tell us, right?
doesn't tell us protein levels.
Correct. Because we can make the assumption, or they're making the assumption that if you put in siRNA for FASN, for instance, you're going to get less of the protein FASN, right?
Hmm.
So our job is to see if is to like fact check or whatever, right? So just because you have FASN siRNA that's theoretically supposed to knock down that, uh, we can't say for sure or for certain that we have less FASN if you have FASN siRNA. So yeah, you would want to evaluate the protein levels of these things to see if the siRNA is indeed knocking down, you know, that the that gene, right?
Hmm.
Okay. So that would be good. And what about two?
Evaluating the siRNA specificity for fast-sound MCAS by testing the cellular levels of unrelated proteins.
Yeah.
Um, yeah, I guess you could include that.
Yeah, I guess you can include that, or you have to include that in order to validate this.
Um, thing is, they're unrelated proteins,
Okay.
but if they're unrelated proteins, like they're not important to our study. So,
So, so what?
hmm. It says specificity.
Yep.
So,
Hmm.
I guess I would include it.
Yeah, right. Because what if the empty Cas siRNA uh just blocked, like, let's say the gene that makes, I don't know, the gene that makes like hexokinase. If the siRNA does that, we're probably going to have reduced cell viability, right?
Hmm.
So so if you checked for an unrelated protein like hexokinase, and we see that, let's say this is hexokinase, and you see it's the same level here as empty Cas siRNA, then maybe the reason the cell viability decreases is because the empty Cas siRNA also silences the gene for hexokinase, right? So if we put an unrelated protein like hexokinase here, and we see that the levels are normal when we add empty Cas siRNA, then we can say, yeah, empty Cas siRNA is specific to empty Cas because it leaves the unrelated protein, in this case hexokinase, alone,
Right.
right? So yeah, these types of questions, like you really have to like stop and think and think, like in terms of what are like the limitations of the stuff of the experiment, and then how can we, you know, validate that. So okay, and then Roman numeral three.
Evaluating the siRNA specificity by showing that a specific siRNA
Uh,
has no
Non-specific.
non-specific siRNA has no effect on the protein levels of fast and empty Cas. Validating the siRNA specificity by showing that a non-specific RNA has no effect on the protein levels of fast and empty Cas. it says a non-specific siRNA.
Mhm.
So like it has no effects. I mean that makes sense because it's not fully specific, so valid. Yeah.
So this is like saying, let's say that we have an siRNA that's non-specific, right? Like for that, let's say instead of so non-specific could also mean let's say it's specific for hexokinase. And if the one for hexokinase affects the amount of MTCAS or FASN, then we can say that, hey, the siRNA, right, uh, that is non-specific to FASN and MTCAS can affect levels of FASN and MTCAS, right?
Repeat it, please.
Oh yeah. Uh so let's say that you put a non-specific, like an siRNA that doesn't silence FASN or MTCAS, right?
Mhm.
We would still want to see if it's supposed to have no effect on the protein levels of FASN and MTCAS, right? So that can be a way for us to see that maybe the levels of FASN and MTCAS could be going up just because of some non-specific siRNA. So I'm thinking like let's say you had a hexokinase siRNA. It's non-specific to FASN or MTCAS, so it shouldn't have any effect on FASN or MTCAS, right? But if it does, then we know that this experiment like could be due to a different uh siRNA.
So you're saying that this it's important to include step three?
Hmm. Because so like okay, step two and step three are very similar, right? But we're switching two different things here. So Roman numeral two, you're using FASN siRNA or empty Cas siRNA to, let's say, see if it has an effect on an unrelated protein. And let's say that that's what I wrote here, hexokinase, right? So if you use empty Cas siRNA and you see the amount of hexokinase decreases, we can then say whether that empty Cas siRNA is specific or not, right? Because if it lowers the amount of unrelated proteins or affects them, then it's not very specific, right?
Okay.
And then Roman numeral three is like the reverse, where instead of using siRNA that's specific for an unrelated protein, we're using siRNA that is non-specific to a related protein.
Okay.
Okay. So two is like, okay, is the MTCAS siRNA gonna just work on MTCAS, or will it affect unrelated proteins? If it affects unrelated proteins, it's not very specific.
Mhm.
And then Roman numeral three is if you have a non-specific siRNA, it should not affect the levels of FASN and MTCAS. But if it does, then that could be an issue.
But what is non-specific siRNA that like
Just think of it as something that is not specific to FASN or MTCAS.
okay.
So so Roman numeral two is you have a specific siRNA, but unrelated protein. Roman numeral three, non-specific siRNA, related proteins.
Mhm.
Mhm.
Yeah.
Yeah, this is like a content kind of thing.
So yeah, here As for metabolic reaction, um, which is affected by treatment of cells with C75. I, for some reason, just thought that it had to do with the mitochondria, so I just picked fumarate and malate.
Okay. So there's something that we wrote um in our proportionalities that can help us answer this. Um, and it's something that is otherwise not important. So,
Cell viability?
oh no, that's important. Um,
Wait, not important? Like,
I
what do
guess
you
like
mean?
not important
This
for
question?
anything other than like this question.
Oh. Long chain fatty acids?
Well, it'll be the lipoic acid.
Oh, that's like super low yield.
I know, I know.
I know lipoic acid is used as a cofactor in fatty acid synthesis, right?
Oh, so it
Or the opposite.
in the pyruvate dehydrogenase complex.
Oh, okay, yes.
Yeah.
Then so I messed it up with biotin. Biotin, is that used as a cofactor in fatty acid synthesis?
Yeah, yeah.
Okay, got it.
Hmm.
Yes, lipoic acid, then it'll be C.
Yeah, yeah. So yeah, this is a
Got it.
annoying low yield kind of question, but
And then I got 96 correct and I understood it, so just this is left. Um,
oh yeah, cool. I always like
so,
to talk about protons and electrons. So,
um, yeah, as based on information passage, lesion empty cast results in decreased level of. I eliminated I um eliminated C and D
mhm.
because I was thinking that it talks about protons.
Yep,
Um, and yeah, then it was just between A and B. I
yep.
was really in between that. I mean, before. Now that we spoke about it more, so yeah, I mean, you have the inner and outer mitochondrial like matrix.
So we know
Uh,
Cas.
what?
Oh, so we know empty Cas is proportional to MMP.
Yeah, mitochondrial memory potential. So
Yep.
that means that like when there's less of MTCAS, there's less mitochondrial memory potential.
Mhm.
So yeah. Um,
Yep. So,
so yeah.
how about this? With empty Cas, which of these would have um a decreased level of protons if you had empty Cas working?
But the thing is, if you have MTCAS, you have more protons that are in the on the outside, but then they end up coming through
Uh-huh. They can end up,
back in.
yeah, they can end up coming back through just, to make that ATP, but it's not gonna make the inside, uh, like the inside is still gonna have less.
Oh, really?
Yeah, because the ETP is
Oh.
still pumping them out. So, because,
Oh, okay. Yeah, it'll be decreased in the mitochondrial matrix.
so now a deletion in empty Cas, right?
Wait, so there will be
Mhm.
decreased amount in the mitochondrial matrix.
Yeah, if you had a working empty Cas, right?
Yeah.
Mhm. So now if you delete it,
Hmm. There's less in the intermembrane space.
yep, that makes sense, right? Because empty Cas makes that is proportional to the membrane potential, and membrane potential is essentially the electrochemical gradient
Hmm.
and inversely proportional to membrane permeability.
Okay.
So normally the empty Cas would put the protons in the intermembrane space, and less of it in the matrix, but then when you mess up MTCAS, you get the opposite, and therefore a lower amount in the intramembrane space.
Mhm. Okay.
Yep. So yeah, that was a good passage because you did a lot more of the talking, and then you said really great things like leads to causal and you're
Mhm.
great job there. So we could have gone through 20 questions, but going through the three or four that we just did right now is way better because you are the one thinking and you're, interpreting things and you're thinking out loud and all that good stuff. So great job. And let's see, we are meeting next week on Tuesday next week, right?
Yeah.
Okay. Um, oh, and
I owe you 175 for Monday.
yeah. And then you could do like 75 for like today. And then uh about the oh yeah, so yeah, for the full length. So like yeah, I don't have much data on the Jack Weston ones, but I mean honestly, it's not like don't take whatever score you get to heart. Like don't be too uh dependent on or don't don't think of it too much. Just know that every time you're taking a test, you're practicing, and every time you're reviewing a test, you're learning, and things that you learn may not show up again for a good many tests. And also, yeah, for Jack Weston, just from like the questions that you've shown me, seems to be on like the tougher side. So bear that in mind. How much would it cost for you to get the rest of the blueprint tests? Because if it's over $100, like it's not gonna be worth it probably. But
So here's the thing. I could get all the Altius exams right now for $100 if I want.
yeah, do it. I think that would be way better.
Yeah?
Yeah, because yeah, that's the stuff that I have like the data from. That's the stuff that like
Really?
yeah. I shared them with you, right?
Yes. Yes, you did.
Altius and Blueprint. Those are like the ones. So yeah, if you could get all 10 for $100, that's super that's a great deal. Um, because the fact is that your Blueprint scores are higher and Blueprint is supposed to be deflated. So and yeah, I just don't know. Like I don't have I don't have data or experience much with the Jack Weston tests, but from the questions that you were showing me on that last test, it just seemed like extremely like the questions you were doing like extremely ambiguous. Like I didn't know there's so much of it that was you know yeah. So I think yeah, get the Altius and do an Altius test on you're doing it on Saturday, right?
Yeah. Here's the thing though. I will say it takes me at least 2 days minimum to review it full length. And like I am scheduled to take the AMC full length too, which, you know, holds great weight in terms of my plan for the next month or two. Like I don't know if I'm gonna push my exam or not. I will only know until I take this full length too. And I'm just wondering the cost benefit analysis of like taking an Altius and then reviewing it for 2 days, which is great. But instead of those 3 total days on Altius, I could spend on section bank and maximize and see how well I can really do on this AMC Falling 2 next Saturday, you know?
Well,
Like,
we're gonna be yeah, so in between, so you're gonna be taking tests like on weekends, and then during the week, that's when you can do your section bank and your stuff. You know what I mean? So
Hmm.
I think that like because what are like what are you going to be doing during the week, right? For the 5 days.
Nothing.
So so section bank you can work on during the week, right? And yeah, and keep doing the tests on, the weekends. Um and yeah, you'll end up doing a lot of the tests and you'll end up, finishing the section bank because, that's what you'll be doing on those like 5 days. So,
Okay, so I'll see us full length one Saturday.
yes, and for cars, you can use AAMC like from like the question packs and stuff.
Yes.
And okay, okay. Oh, and then yes, remember not to so I mean like this can sound like a lot of repeating, but read the entire passage, okay?
Hmm.
No skimming, right?
Yeah.
Okay. Make sure you read the entire thing, no skimming, and yeah. Um
And lastly, for the AMC, here's the thing. When I take a full length, um, and there's 9 cars passengers, which is a lot. Like it's kind of hard for me to blind review every single one. So what I'm thinking, like every single cars passage blind review the next day, all in one day, plus all 3 other sections. That's like a lot. Like I some people can do that. I just am not built like that. So what I'm thinking is after like let's say Saturday, take the AMC full length, the next or whatever, the Altius plus cars, whatever. You get what I'm saying. The next, I guess, 7 days or 6 days, like every day I would do one cars passage from the
let, let,
AMC.
let's just do this. When you do your blind review for cars, just choose two passages in which you've gotten the most questions wrong.
Okay.
You could just do that.
But then how about the other 7? Like I that's just like leaving stuff on the table, like
Well, if the alternative was to not do that and then doing like the cars stuff that you were just telling me, then I think because you were just telling me like, instead of doing like the blind review for all the cars passages, that you will do like the cars every day at some
but
point.
I would do like because Jack Weston has you do it twice, right?
Mhm.
Like what I'm saying is I would do the each of the cars passages I took on the full length, I would do one each day, like a second time, and then go through them.
Oh,
That's what I meant.
and okay, what? And then you said when was this? This was like at some different point in time.
No, this was, I think you're misunderstanding what I'm
Yeah.
saying
Sorry.
because I'm also being confusing, but it's fine. Okay, I get what you're saying. I'll do that. No, what I'm saying, what I meant is like instead of doing a new passage, I would just redo the ones from the AZ one the second time in the next couple days.
Got it. Okay, so the blind review of cars is just you're just gonna
Uh,
replace the regular doing a new passage. Okay, yeah.
yes, yes.
All right. Yes, you can do that.
Okay.
So yeah, remember read the entire thing and remember to if you get stuck and like a math problem or something like that, guess, flag, move on, and good luck. Okay.
All
Awesome.
right.
Awesome.
Yes.
All
I will
right.
see you.
All right. Take care.